Hello everyone, welcome to MMBS111 Introductory Microbiology. In this mini lecture we're going to be talking about methods of physical control of microbes. So this mini lecture will address concepts from section 9.2 of chapter 9. There are 6 learning outcomes that you need to master by the end of the section. The first is name 6 methods of physical control of microorganisms. 2, discuss both moist and dry heat methods and identify multiple examples of each. 3, define thermal death time and thermal death point. 4, identify advantages and disadvantages of cold and desiccation. 5, differentiate between the 2 types of radiation control methods. And finally, explain how filtration and osmotic pressure function as control methods. So first we're going to talk about heat. Heat is probably one of the most used methods of physical control. Elevated temperatures above that maximum growth temperature are microbicidal. This means that microbes are killed. And hopefully you remember from chapter 6 that each organism has its own temperature range of preferred growth. And once we get above that maximum growth temperature, then certain adverse things begin to happen, like denaturation of proteins and those kinds of things that make it impossible for the microbe to continue to function properly. Lower temperatures, or those temperatures that are below the minimum growth temp, are microbistatic. This means that, if you remember from the previous mini lecture, that the microbes are not going to be reproducing. We are not going to increase the population. Okay, so static, the population growth has ceased. Okay. Moist heat examples include things like hot water, boiling water, or steam that has reached the temperatures between 60 and 135 degrees Celsius. When we're talking about dry heat, we're talking about hot air or an open flame, and those temperatures can range much higher, significantly above 160 degrees Celsius. So moist heat typically operates at lower temperatures and involves shorter exposure times to achieve the same effectiveness as dry heat. The microbicidal effect of moist heat is that the microbial proteins are coagulated and denatured, and this results in death of the organism. Dry heat at sort of lower temperatures dehydrates the cell. It removes the water that's necessary for all of those metabolic pathways. At a little bit higher heat, it can denature protein. At very high temperatures, typically an open flame like the Bunsen burners in the lab, this is going to oxidize cells and turn them into ash. There is a variety of levels of heat resistance depending on the organism that you're looking at. So bacterial endospores tend to be the most resistant to heat. Destruction of spores usually requires temperatures well above boiling, and resistance is going to vary depending on the spore. Vegetative cells are the least resistant to heat. Death times are going to vary from 3 minutes at 50 degrees Celsius to 60 minutes at 60 degrees Celsius. So it really just depends on the cell type. Viruses have varying heat resistance. So it may take 2 to 5 minutes at 55 degrees Celsius all the way up to 600 minutes at 60 degrees Celsius. So there's a wide range of heat resistance among viruses. When we're talking about heat and death of organisms, we need to address these two different points. So thermal death time is the shortest length of time that's required to kill all test microbes at that specified temperature. The thermal death point is the lowest temperature required to kill all microbes in a sample in 10 minutes. So each of these can be used to evaluate the effectiveness of certain heat control processes. So we're going to look at first moist heat methods and the different methods that use moist heat in order to kill organisms. So the first and probably most familiar is boiling. 30 minutes of boiling will kill most vegetative cells. The unfortunate part of boiling is that the object that you have boiled can easily become recontaminated after boiling, and the material has cooled. Typically, boiling is only used when we're trying to disinfect water or other inanimate objects. Notice that boiling is not a method of sterilization. It is only a disinfectant. Pasteurization is another method that uses moist heat, and it kills the potential agents of infection and spoilage while not damaging the product that we're trying to disinfect. So it is not sterilization. Many microorganisms will still remain. That will eventually lead to spoilage. Typically, pasteurization is used for the disinfection of beverages like milk, some alcoholic beverages, fruit juices. The final process that uses moist heat is autoclaving, and this is steam, high temperature steam under pressure. And the pressure is controlled and increased by creating steam in a chamber that can then be pressurized. And that increased pressure also increases the temperature. So 121 degrees Celsius in the autoclave is hot enough to sterilize materials. And autoclaving is typically used to sterilize tools and equipment that can withstand the moisture and the heat. Any culture media that you're going to use in the microbiology lab, surgical dressings, paper, and some plastics, depending on their composition. We also use autoclaving to sterilize waste before it is disposed of into the environment. Dry heat methods, there are two methods that use dry heat. Incineration, and this is the use of an open flame, either a Bunsen burner, like you've hopefully used in the lab, or an incinerator. And these are both used to reduce microorganisms to gas and ash. So we're essentially burning them up. This can be used to sterilize lab instruments and contaminated materials like dressings, bedding, carcasses. The hot air oven is another method that uses dry heat, and air is circulated in the oven at 150 to 180 degrees Celsius for 2 to 4 hours. And this method can be used to sterilize glassware, metallic instruments, and then powders and oils that cannot be sterilized in the autoclave because of the moisture. We can use dry heat to sterilize those substances. So the opposite of heat, of course, then is cold. And cold treatment slows the growth of organisms, but it does not kill organisms. So cold treatment is bacteriostatic. It slows the growth, so they're not reproducing. It's an effective method to reduce contamination in foods. It helps to extend the life of food products. But most microbes are not going to be adversely affected by cooling, long-term refrigeration, or freezing. In fact, this is how many organisms are preserved for later study. Temperatures from -70 to -135 degrees Celsius can preserve cultures of bacteria, viruses, and fungi for long periods of time. And then they are gradually, defrosted, warmed up, and then we can grow those organisms again later on. So desiccation involves the removal of water. So essentially the organisms are exposed to room temperature. The water evaporates over time, and they become dehydrated. Some microbes are killed by this process, but most microbes are not affected by desiccation. We use desiccation in lyophilization. So lyophilization is the combination of freezing and drying, or desiccating. And we use this to preserve microbes so that they can be studied for many years. This is also how microbes are sold in the microlab. So if you need a particular control organism, or there's an organism you want to study, you can actually purchase lyophilized bacterium. And once you expose those organisms to water and proper nutrients, then they're able to grow again. Radiation is another physical method that is used to control microorganisms. When we're talking about radiation, we're talking about different wavelengths of light, and depending on the intensity of those rays, we're going to get varying control. So ionizing radiation uses gamma rays and X-rays to control microbial growth, and this actually results in the sterilization of whatever is exposed to that ionizing radiation. Generally, we're going to reserve this for substances that are sensitive to heat. And that involves things like vaccines, drugs, some plastic instruments, surgical gloves, any tissues. After the anthrax scare in the 90s, mail is now, well, some mail, depending on its destination, is also exposed to ionizing radiation. And some foods as well is exposed to this type of radiation. Non-ionizing radiation involves the use of ultraviolet rays or UV rays, and this results in disinfection. And generally, we're going to reserve this for the use on certain gases and liquids. There are germicidal lamps that are installed and used in many operating rooms, the lamps used in dental offices, certain food preparation areas use these lamps, and they help to disinfect the air and the space. They can also be used to disinfect water and some other beverages. So there are some non-ionizing radiation tools that we use to disinfect our drinking water. Filtration is another physical method that is used to control microbial growth, and we can use filtration on fluids and air. Fluid filtration can result in sterilization if the pores in the filter are small enough that no organisms are able to pass. And you can purchase filters with different pore sizes depending on the organisms that you're looking at. So generally 0.02 microns that will address the smallest organisms that can be found in fluids, and that would result in sterilization. The only drawback to fluid filtration is that it's not able to remove the toxins that can be produced by some microorganisms that result in disease. Generally, we use filtration when we're trying to sterilize fluids that are heat sensitive. So vaccines we can use filtration for. Serum and blood products, IV fluids, enzymes and media, we're going to use fluid filtration. Air filtration results in disinfection of the air, especially if you're using a HEPA filter, and these are typically used in hospital filtration systems and other clean room situations where you want to remove as many organisms from the air as possible. And our final mechanism for physical control is osmotic pressure. And we learned in chapter 6 that organisms are very sensitive to osmotic pressure and differences in solute and water concentrations in the environment. So adding large amounts of salt or sugar to foods creates a hypertonic environment for bacteria, and this can cause plasmolysis. So remember, in a hypertonic environment, the water is going to leave the cell and enter the environment. This is going to cause the cell to shrink, and that's plasmolysis. Pickling, smoking, and drying foods have been used for centuries to preserve foods. We still use these methods today in food preservation. However, this is not sterilization, so organisms will remain. We're not going to get rid of every organism by using any of these methods. And that is the end of mini lecture 9.2. These are some of the checking in questions. Make sure you go through these questions, that you can answer them correctly. Remember to go back and review the learning objectives for this mini lecture, and be sure that you have mastered those concepts. Here's our critical thinking question. The answer to this question is on the next slide. So if you want to work through this and then go back to the video or the PowerPoint to check your answer, that would be a good idea. So our question is, Jennifer purchases a half gallon of milk from the grocery store. 2 weeks later, Jennifer opens the carton and pours some of the same milk on her cereal. Unfortunately, Jennifer realizes that the milk has gone bad. If the milk was pasteurized, what has caused Jennifer's milk to turn sour? All right, so make sure you go back and check those out. Hopefully this video was helpful.